Hi there, my name is Konan Kravchuk, and today I'm going to talk about antibody sequence liabilities. So altogether, sequence liabilities and antibodies are a very important topic, and they are mostly tackled using, let's say, like quite basic computational techniques. Yeah, so today I'm going to talk about what those sequence liabilities are from more like a chemical perspective, but I'm going to put most weight towards the computation analysis of those. So a sequence liability is any kind of chemical reaction that could make your antibody store bad on shelves, make it less manufacturable, or have undesirable in vivo effects. And mostly they come about by very specific amino acid motifs, yeah, that are either two or even like in three amino acids long, but like some individual amino acids are quite susceptible to such modifications as well, such as tryptophan or methionine. So let's have a look at one example of a sequence liability so that we actually see like in what can happen in your antibody. So assume that you have asparagine, and this asparagine can have its side chain modified by the interaction with the backbone. And it can change into either isoasp or aspartate via an intermediary of succinimide. And of course, this is not desirable because it can change the local structure, which can affect the binding. And in terms of manufacturability, it can affect the shelf life of the antibody. So altogether, you want to make sure that such modifications do not occur. And one way to mitigate it is make sure that the said asparagine is not going to be next to certain other amino acids, like the small one, glycine or serine, where it can actually interact, where the side chain can interact with the backbone. So what liability motifs are out there? Well, this is something where we started looking at the literature, the organizational knowledge, and altogether, most of this knowledge is actually accumulated in silos in organizations. Yeah, like people are moving across companies, so like this knowledge spreads around. But altogether, we have not found something like, let's say, like a textbook reference for antibody sequence liabilities. So as part of our analysis paper, so lab of sequence liabilities, we actually created a reference of our own with our colleagues. So what you need to know is that there are specific liabilities. They're associated with certain motifs that are either like one, two, or three amino acids long, and they have assigned severity to those. So it is not to say that those severity, like medium or severe, like are written in stone, like and they can change between organizations, but some of them actually make sense. So, for instance, if you have an unpaired cysteine that can engage in a covalent bond, then this is, of course, quite a severe liability to have. So now moving on towards the computational identification of those. So because those liabilities are typically just, one, two, or three, motifs, like, motif of one is actually, of course, not a motif. I always thought of that more as a glorified spell check for antibodies, because in many cases, people just can have those liability checks literally in Excel. Yeah, like you have an antibody sequence, and if you see NG or NS somewhere in your CDRs, then you just can flag it, and based on your engineering knowledge, you might either want to engineer it out or not. So here comes the question, yeah, like what is the engineering intuition towards engineering it out or not? Because it is not a given that a given sequence liability motif is actually going to materialize as a chemical modification. So now we're going more from the analysis and identification towards prediction. Yeah. And the question is, can you actually predict whether there is going to be a sequence liability in general, or rather a chemical modification? And the answer to this is, of course, not. Yeah. because those are extremely dependent on your manufacturability conditions, formulation, and storage, and so on. Yeah. Notwithstanding the in vivo conditions that are quite difficult to check anyway. So if you want to have a proper predictor for if you have a sequence liability and whether this is going to incur a chemical reaction, then you would need to generate your own internal data according to certain conditions and then have a predictor based on that. So the other question is, should you not bother with sequence liability analyses and prediction at all? Of course not. Like, it is still a problem, and it is important to build an intuition and a toolkit to actually be able to tackle those. So one part that we have done in our lab study is that we actually want to see how prevalent those sequence motifs are that can lead to sequence liabilities. So you know that we have collected quite a lot of data sets from the public domain, so everything from GenBank, next generation sequencing, therapeutics, patents, literature, across the board. Yeah. And we looked at our liability reference and essentially counted how many of those there are in different sequences. And my hypothesis was that in therapeutics, especially the newer ones, you're just going to have less of those. Yeah, perhaps in natural antibodies, you're going to have more. But lo and behold, actually, all of those sequence liabilities are actually quite prevalent in quite equal numbers. Yeah, I'm saying equal numbers, like in this graph, the NGS and therapeutics actually have two chains. So, and all the other data have one chain. So actually, this puts them at parity. So actually, the sequence liabilities across sequences, if you just count the motifs, are present in pretty much similar numbers. Yeah. So this essentially means that we're dealing with with a problem of false positive identification. Yeah. So because we could not build a predictor that is general towards telling whether a given chemical modification is going to happen, because I told you that this is impossible, we have gone towards identifying whether something can be more innocuous than your next liability motif. So we have looked at what is out there, and quite a lot of this work was actually condensing what is out there rather than coming up with stuff on our own. So we have come up with 3 liability flags, and the first one is the germline presence. So the intuition for this is if it's present in the germline, then at the very least, nature does not mind it being a heterogeneous binder, yeah, in vivo. So, but this has nothing to do with manufacturability, yeah, because we actually did not evolve antibodies to be manufacturable, unfortunately. Next flag is occurrence in therapeutics. This one actually Amgen came up with, and the intuition here is that if you have seen a given sequence liability in a marketed therapeutic, then it has gone through all the hoops, clinical, preclinical, and so on, and it is fine. Yeah, so that's it. And the last one, the surface accessibility. Here, the intuition is that if you are buried, then there is less of a chance that you are going to have a chemical modification that is actually going to be detrimental for the antibody. So we have those 3 flags, and we would like to say that if you have a certain flag, a motif with a certain flag, then perhaps then this could be a false positive. Yeah, of course, this is our hypothesis, and I'd like you to actually see that this is actually helpful. We need to answer 2 questions. One, do you actually reduce the number of sequence liability motifs that you'll be looking at? Yeah, because if you have a sequence and you apply the motifs and you have 10 of them and you still have 10 of them to look at after applying the flags, then it's not very useful. The other one that is much more difficult is actually ensuring that those liabilities that are flagged are less of a risk to be actually detrimental chemical modifications. Yeah. There aren't that many data sets out there, but we found some, so we actually did some validation here. So on the first count, where they're actually reducing the number of liabilities comes from applying those flags. That's the case. So this is the liability motifs that I've shown you before. On the left, you see all of the liabilities if I don't apply any flags. And after I apply the flags, then these are all the motifs that are left that do not fit the bill of either being on the surface, on the germline, or in the market therapeutic. Yeah, so you get to look at pretty much half of what you used to. Yeah, which is already good. Yeah, like this is less work. Next one, this is much more difficult, was to actually see whether you are reducing the number of um, uh, liability motifs that could actually be detrimental. Yeah, because if I take away a motif that is, say, like on the surface or a germline or therapeutic, but actually it is the problematic one that you should have looked at, then it's not a very useful tool. So we looked at data from the paper that I'm studying here below, and they have actually generated experimental data for antibodies with deamidation and isomerization modifications. So for each of those graphs, on the left, you have the total number of liabilities that they have actually seen. And towards the right, you see what is the proportion of seeing, or actually counts, of the modifications within certain filters. Yeah. So clearly, like if you have a certain number of all liability motifs, you would hope that those having a certain flag would actually be a smaller proportion of those, yeah, because otherwise it is not very useful. So in all the cases, you can see that the number of liabilities that are fitting one of our filters is actually less than the total number of liabilities, which is actually pretty good, yeah. Although, like, I would take it with a grain of salt, yeah. So going towards conclusions, essentially, if you were looking for like analyzing sequence liability motifs, do check with your organization. Like we do have a reference, but do check what severity levels and what motifs your organization is looking at. I would use the predictions mostly as a false positive flag, not as a very definite prediction that like this chemical modification is going to happen. And if you want proper predictive power, I would strongly suggest that you generate your own data, but under very strict manufacturability conditions. So finally, where can you actually perform such analyses and annotations of sequence liabilities? Honestly, quite a lot of this work happens in Excel still. And if you just want to define certain liability motifs in Excel and use that, I'm pretty sure that you're going to be fine. But if you want to do it at a scale, then normally like I would cite a lot of like a good academic work, but actually like there aren't that many tools to do this precise task. Yeah. So it's either enterprise grade antibody analysis software, or of course, tooting my own horn, you can use our own platform, which is for free. So here, using the sequence and engineering module, you can see for a single sequence where such liability motifs are. You will see our false positive flags for them. And you also get a lot of other options to help you engineer this modification out if you decide that this needs to be done. In the batch sequence annotation module, you can annotate a lot of those sequences and then perform large-scale filtering by if you don't want exposed cysteines, or if you essentially do not mind certain sequence liability motifs. Finally, you can also see those modifications from a structural standpoint if you log into the structural modeling module and look at the liability annotations over there. So here it is essentially a shortcut of like modeling your antibody structure and seeing the liabilities right away without having to do the scripting. That's it. Thank you very much. I hope that you found it informative.